Yeah, okay. So it's 6:03. I think I'll start now. So yeah, hi all. Welcome to my TA class. Today mostly we'll have a introductory section. So as we proceed further, we'll discuss in details. So today it is not going to be that difficult. Fine. Let's see how many of you have joined. Yeah, can anyone just confirm? Am I visible? Am I audible properly? My voice is fine.
Yes, sir.
I couldn't hear you, Aishwarya. Were you saying something? Yes. Okay,
Yes,
great.
sir.
Okay, so yeah, I have given you access to the mic. You can turn on your mic and ask if you have any question. Uh, like if you don't have any question, please prefer to keep it off to avoid any disturbance. Okay. So yeah, I think it's 6:05. Then we'll start now. So I'll be discussing. So my class goes like this. I discuss some concepts. I tell you
Yes,
some
sir.
basics, some theory, and then I'll solve some questions. So it's like I'll be. I think you have week zero, which is your prerequisite questions. You have your week one, and you have your week two, like now. I'll be discussing some theory related to week zero questions. So in my class, it's not like you remember everything and you give the answer. It's more like how you come to the answer. So please add my friend. Actually, the meeting is open to everyone. Everyone can join. If you click on the link, please forward the link to your friend. I think he'll be able to join then. If you want, I'll put the code here. So this is the code. Just tell your friend to put this code in Google Meet and then he'll be able to join. It's open to all. Okay, great. So yeah, today it's not going to be that difficult. Uh, by the way, you people are undergraduates, right? Or like UG level, or you are graduate students. You must be BTech or BSc students, right?
Yes, sir.
I, Sorry, I think you're speaking something, but your voice is not at all audible, or my volume is down. Okay, let me check. Okay, okay, let me put my phone also in case you need to type anything in the chat box. So yeah, if you all are comfortable, let's start today's session. So I think some or 16 people are here. Okay, I'll put my slide now. Okay. So I put my PPT. Can just anyone confirm for the last time? Am I audible and my PowerPoint is visible to everyone?
Yes, sir.
Okay,
Yes,
great.
sir.
Okay, okay. So hi all. I'll start with the basics of biology, week one problem solving session. I'll be discussing some concept. As I said, today's will be very introductory session. So most of the concept or the things I'm going to tell you, you have read in the biology of your class 11 and 12. Uh, so without any further delay, let's start. So this is as this is the very first TS session or the first week one from my side. I have to start by a basic introduction about me. So I'll give a very quick introduction about me. So I'm Rajesh Das. I am a PMRF research scholar currently in the Department of Biotechnology, IIT Madras. And I did my MTech in Biotechnology and Biochemical Engineering from IIT Kharagpur in the year 2022. And I did my BTech in Biotechnology from NIT Rourkela. So after my MTech, I was in that summer school exchange program at the Friedrich Schiller University, Jena, and the Jena University Hospital in Germany before I joined here in IIT Madras in 2022. So I'm currently a research scholar and I'm working in cancer biology in Professor S. Mahalingam Sir's lab in the Department of Biotechnology, IIT Madras. So that's all. You have my email ID here in case you want to reach me out for any question and queries. So let's dive into the course. So on your left side, you can see there's a very dense forest. So if you were to visit this dense forest, you would actually see a great number and kind of living organisms in it, varieties of living organisms. Each different kind of plant, there will be plant, animal, or let's say so some other organism that you see. Each of them represents a species. So this is all known to you. What is a species? So whatever you see, what kind of organism you see, let's say in this dense jungle is called a species. And it is like approximated that the total number of species that are known, it ranges between 1.7 to 1.8 million. I hope you all know how many zeros are there in a million. Okay. So it is 1.7 to 1.8 million of species are yet discovered. There are the list goes on. People are discovering new species, new organisms every time. We will talk about that. So this is so much or the so much of species. Actually, this refers to biodiversity, or what is in simple term, what is biodiversity? It is the number and types of organism present on Earth. We should remember that as we explore new areas, even the old ones, new organisms are continuously being identified. So the list is not a constant or a static list, but the list goes on. Like people keep on identifying new species. For example, there are species yet to be discovered and are being discovered very deep down the ocean or like say a hot spring. Uh, for example, if you had read about the PCR, so PCR, the enzyme is Thermus aquaticus, that from there they extracted the enzyme, so it is also active at very high temperature. So my point is, you keep finding organisms or different species in areas that you have not even expected to find there, and the list goes on. So my second thing would be how this much of diversity was created, or how come this diversity was started. So it must have started from something. So if there is so much of 1.7 to 1.8 million of species, then it would have started at some point, or some point is zero. It should have started from zero. So that we are going to discuss origin of life, how from this zero everything started. So you must have heard about the Big Bang theory that I'm not going to discuss here. In this, I'll start from directly from the solar system of our Milky Way galaxy. So, in the Milky Way galaxy, Earth was supposed to have been formed about 4.5 billion years back. So, billion is 10 to the power 9. Okay. So, it is 4.5, it is estimated or that This is approximate age of the Earth, 4.5 billion years. So these two scientists, Oparin of Russia and Haldane of England, they both proposed that the first form of life could have come from pre-existing non-living organic molecules. Note this point: pre-existing non-living organic molecules, that is RNA and protein. So how this big molecules, that is RNA, protein, came into the picture, that is thing to think. Uh, we'll also see how these protein RNA might have come into the picture. And the formation of life, what they also told, that the formation of life was preceded by chemical evolution. So first happened was chemical evolution. From chemical evolution, this non-living organic molecules formed. From this organic molecules, actually, the cells originated. So the condition on the earth, so when the earth formed, it was a very hot mass. I think you all have read about this in physics, that there was high temperature, there was volcanic storms, there was a reducing atmosphere which contained ammonia, methane, so CH4 is methane, NS3 is ammonia, etc. This is the initial atmosphere or initial atmosphere of the earth when it was formed, and it was reducing atmosphere. So one scientist in 1953, his name was S.L. Miller. He was an American scientist who created similar conditions in his laboratory. He created electric discharge. So electric discharge in the on the right side you can see it. It resembles the continuous lightning or thunderstorm that were continuously going around, or probably during that time. So it mimics the continuous electric discharge, and this closed flask was to mimic Earth, containing the same gases or the same reducing atmosphere, methane, hydrogen, ammonia, and water vapor. Of course, water was there first. Water vapor at 800 degrees Celsius, because that time the temperature was very high. And interestingly, when the so here the reaction was carried out. Here the gases were there. Here continuous sparks were there, and then as the liquid cooled down, it flew or it flowed down, and here it was collected. And when he analyzed what all has been formed and what is there in this water, he observed that there are amino acids. So, if you know, amino acid are the building block of protein. So, thing is, it started with methane, H2 or hydrogen, ammonia, water vapor, and interestingly, some chemical reaction occurred. These all were inorganic things. From inorganic things, amino acid, which is organic, was formed. So there was chemical evolution or chemical reaction, from which the inorganic components turned into organic components, and from organic component, the first cell or the first moving cell or the living cell would have originated. That is a debate still going on, how like the initial or the pre-existing cells would have originated. Some theories, DNA, RNA, and protein would have come together, and like DNA won't be there initially, only RNA would be there. RNA and protein would have come together, and it would have formed the first bacteria, like not modern bacteria, very pre-existing bacteria. This eukaryotes would have very pre-eukaryotic eukaryotes would have been formed. Then from some advanced eukaryotes had been formed. So from prokaryotes to eukaryotes have been formed. Like that, life would have originated. So I hope till here I'm clear, and you people are able to listen to me properly. Just confirm once, because I can just see my PPT, and I can see the chat box.
Yes, sir.
Okay, great.
Yes, sir.
Okay. So that is all about the origin of life. Okay. So as I said, the first cellular form of life did not possibly originate till 2 billion years ago. They were possibly single cells because it would have taken some time for the chemical reaction to occur from inorganic components to give rise to organic components. And they estimate that the first cellular form of life did not possibly originate till 2 billion years ago. There were probably single cells. So initially, only single cells would have been formed. All life forms were in water environment only. This version of biogenesis, that is, the first form of life, arose slowly through evolutionary forces from non-living molecule, is accepted by majority. This is what the global scientific community accepts, that the first form of life slowly arose through evolutionary process forces from non-living molecules. It's not the alien concept or something that spores from the sky came and all that. At least that is not being accepted by the scientific community. Okay. So the next is the I talked about evolution. So evolution is something starting with the zero and then going something towards which is advanced or higher than zero. Let's say one, two. So it is starting with nothing and going towards something. So evolution of life forms, it is a theory, and this theory was given by the person here on the right side, and he's Charles Darwin. Okay, so Charles Darwin actually he took a sea voyage in his ship called HMS Beagle around the world and concluded, and he found out that existing life forms share similarities to a varying degree. So the all the existing life forms actually have so much of similarity among themselves, degree among not only among themselves but also with the life form that existed millions of years ago. So he found out, he kind of concluded that he observed so many life forms and like concluded that they have so much of similarity among themselves, but not only among themselves, but also the life form which existed millions of years ago. So something which started millions of years ago or millions of years back is now existing in a different form. So many such life forms do not exist anymore. There had been extinctions, like extinction of dinosaurs, of different life form in the year gone by, just as new forms of life arose at different periods of history of the earth. So there has been gradual evolution of life forms. So these are all the things that Charles Darwin wrote in his paper. Okay. So one more thing. Is that according to Darwin, Charles Darwin, he mentioned a word called as fitness. So fitness, according to Darwin, remember this, he told that fitness is ultimately and only reproductive fitness. But now we know that fitness is not only limited only to reproductive fitness, but also other factors are also there, like uh, how well the organism can sustain or survive the environment. It is somewhat related to reproductive fitness, but not only reproductive fitness, but other types of fitness also contribute in the overall survival of the organism. But according to Darwin, he said that it is only the reproductive fitness. So what he said was that who are better fit in an environment leave more progeny than the others, and these therefore will survive more and hence are selected by nature. And he called this as natural selection and implied it as a mechanism of evolution. So, like imagine this, you have a agar plate. I think you people have seen microbiology experiment where they in a agar plate they grow bacteria. Anyone has seen agar plate where bacteria is grown?
Yes, sir.
Yes. So if you see, like some colonies of bacteria survive, or some bacteria are able to form colony, but not all. If there are like thousands of bacteria, only hundreds of bacteria will be able to form. I'm not going into the very details of how they are forming colonies and all. I'm just giving one example of the selection mechanism or the survival of the fittest. So only hundreds of bacteria will be able to form the colony. Why this hundred of bacteria? Let's say these your plate is ampicillin containing plate, or the selection marker is ampicillin, and these hundred of bacteria are ampicillin resistance, or they have the plasmid which is ampicillin resistant. So naturally they will survive and they will be able to grow on the agar plate and not other bacteria. So from here you can imagine, like these bacteria will be able to reproduce more, and they will be able to produce colonies, and they will be actually fit for survival in that agar plate. So that is one way to think about the natural selection. Okay, so that is a kind of an example of the reproductive fitness or the natural selection. However, the ancestors were present. So, as I said, Darwin told that the life forms he observed had a similarity to the organism which existed millions of years back. So, they are the ancestors. And these ancestors were present at different periods in the history of Earth, like epochs, periods, and eras. They were present long, long back. So I'll give you one example here, like how the organism originated from one. Like let's say there was a ancestor, and from that ancestor or ancestry, how different organisms arised. I think most of you have heard about this. The Darwin finches, Darwin's finches, these birds. So the scientific term I was talking about is called adaptive radiation. I'll just draw a picture about that, but this is the heading, adaptive radiation. I'm going to talk about adaptive radiation. So during his journey, Darwin took his throughout the world, and throughout this journey, he went to this Galapagos Islands. So it is a island west towards the equator. Currently, equator towards the west, they have this island. This is called Galapagos Islands. So you can see it is it has so many islands. It has a lot of islands, and it is completely isolated or segregated from the mainland by a huge ocean present here. So probably there is not so much of communication between the landmass and the segregated or the isolated islands. So let's say one organism or a bird migrated here, it would be very like trouble or a very hectic process to migrate back. Why it would like to migrate back? So it will because there will be a lot of food, lot of space for it to stay there. So there's no point of going back to the mainland. What he observed was that in this Galapagos Island, that an amazing diversity of creature. Of particular interest, he saw these small birds, black birds, later called Darwin's finches, after his name. He realized that there were many varieties of these finches in the same island. So in a particular island, there were different varieties of finches in the same island. All the varieties he said, or he hypothesized, that it evolved on the island itself because it is very far away from the mainland. From uh, from the original seed-eating feature, many other forms were altered, like weak arrow. So, according to him, the initial or the ancestor who migrated was seed eating. So, I'll put a point here. Okay, so this is the ancestor. which was seed eating, and he migrated to one of the island. Now from this, some of the birds migrated to different islands on the like the nearby islands. So let's say where A migrated, there were a lot of seeds. So it has a different kind of beak form or a mouth form that was suitable for the bird to eat seed. The bird, like let's say B, C, D, or E, which migrated to other part of the island, let's say there were insects more and the seeds were less. Now, in order for the bird to survive, it cannot only or rely on or depend on seeds alone. It has to start eating seeds, insects. So gradually, over the period of time, their beaks got modified so that they can also eat the insects. And as you can see, similarly, for other food habit or the other food requirement, their beaks only or their mouth only got modified, but the bird is same. The bird is same, that is this black color bird, but with different kind of beak or mouth based on the requirement of their food habit. Some eating seed, some eating plant maybe, some eating insect. Since it started from one point and then went like this, it looks like some rays of a sun, right? So that is why the name of this concept is called adaptive radiation. Radiation because it looks like rays, and adaptive because it adapted to eat insects and other things apart from the seed. So this is the reason why it is called adaptive radiation. Okay, so I hope everyone is clear till here. Yes?
Yes, sir.
Okay, great. So that was about the. Is anyone typing anything? Just give me a second. Has raised a hand. Yes, okay, yeah, tell me. You can ask questions. This is a TA session. You can ask your questions. Yeah, anyone would like to ask anything? If not, I'll proceed. Oh, okay, if no questions, then I'll proceed. So remember about this adaptive radiation. Okay, so it kind of radiated. That is why it is called radiation. Okay. So based on yeah, these concept, answer these questions. So what was the end product obtained in the Stanley Miller or SL Miller's experiment? Was it protein, amino acid, DNA, or RNA?
Mixture of amino acids, sir.
Okay. Yes, others you can answer or put in the chat box. It's up to you. Yeah, okay. So you are correct. Yes, it is a mixture of amino acids. Yes. Okay. Someone said A. No, the proteins were not there. Protein are a bit advanced thing. So from inorganic things, organic things first appeared. So it was amino acid first, and then amino acid is the building block of protein. So from amino acid, proteins came. So SL Miller first was amino acid. Actually, don't get confused between these two. You may get in your exam and then you might accidentally put protein. No, it's not protein. Remember, it's amino acid. Yes. Okay. Great. Okay. So it is option B. Okay, your second question. The appearance of different species of Darwin's finches is due to migration and adaptation to different location, artificial selection, gene cloning, or insertion of foreign gene.
A option, sir.
Yes. Yes. Okay, others? Yes.
A,
Okay.
sir.
Yes, yes, correct. You all are correct. Yes, as I said just now, it was adaptive radiation. So they migrated to other place. They found different food habit, and accordingly their beak or mouth changed. And that is not a one day process. It would have taken hundreds of years for the beak to evolve. Okay. Okay. So it is option A. So I said about evolution, right? But what is the evidence of evolution? How to, like, in this time or in this particular present day, how can you tell that evolution has happened? So evidence that evolution of life form has indeed taken place on Earth has come from many quarters, and the first and the most important evidence of evolution is fossil. So, what are fossils? Fossils are remains of the hard part of life form found in rocks. So, if you see here, this is the layers of earth, and if you keep on digging this layer, you will find, or you can see the last layer would have been the oldest layer, and the subsequent layers formed above would have been recent layers. So the first layer will be very recent. The layer next or just below this would have been little older. The next one little more older. This is the little more older, and this is the oldest one. So when you dig up these separate layer, they actually people found different forms of life, not alive, but in the form of fossil, like dead remains, bones, or in the picture above here, you can see a mosquito trapped in this is, I think, sapphire or Yellowstone, something like this. So, and when they tried to find the age by carbon dating or something, they found out it is like millions of years old uh, organism. So, fossil is one important part of the evidence of evolution. So, as I was telling, that different aged rock sediment contain fossils of different life forms who probably died during the formation of particular sediment. Sorry. Some of them appear similar to the modern organism, but thing is that the fossil which appear similar to the modern organism, they are most likely or are to be found towards the sediment or the layer which is close to the top, because over time actually the morphology changes or the shape of the organism changes, like you can see here in the third or the fourth layer, this is not very similar to the first layer. So the as I was telling, some of them appear similar to the modern organism, and they will be towards the nearest layer. Like they represent extinct organism, for example, dinosaurs. Study of fossil in different sedimentary layer indicates that the geological period in which they existed. So different geological periods or different parts of the time, they have formed different layers. And by studying different layers, you can figure out about the different geological period and what kind of organism were alive during that duration. So the study also showed that life form varied over time, what I just told now, and certain life form are restricted to certain geological time span. For example, dinosaurs are not yet alive. Hence, new form of life have arisen at different times in the history of Earth. All this is all this evidence. The scientific name is called paleontological evidence. This is the scientific name. So this is about the evidence of evolution, which is fossil. Okay. Okay. So a few more questions based on that. Uh, Archaeopteryx. I don't think if you know about this or not. Let's see who knows. Archaeopteryx is a link between, is it bird and a reptile, bird and an amphibian, reptile or mammal, or reptile or amphibians? So mammals won't be there. We are talking something about old. So it's between bird reptile, bird amphibian, or reptile amphibian.
The
Sir, bird
bird
and
and
reptile.
reptiles.
Yes, 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 correct. So you will know. So it is bird and reptile. So what is this Archaeopteryx is this type of looking organism. Actually, they created this 3D from the bone that was found in Germany in 1861. So Archaeopteryx was a small bird-like dinosaur. It was small. It was very small, and I I don't know if you all people know or not. The birds, the existing birds today have actually evolved from dinosaurs. This is what people have their theory. And this Archaeopteryx, it lived during the late Jurassic period in what is now in Europe. So the discovery of the first Archaeopteryx fossil was in Germany in 1861. Now it has been long accepted that Archaeopteryx was a translational form between birds and reptile, and that is the earliest known bird. This is the earliest known bird. Okay, remember this. Okay, I'll just quickly take a break. Just 1 minute. Give me a minute. This I'll resume now. Keep it just 2 seconds. So when I was, we finished this Archaeopteryx. Oh. So I was talking about the fossils, right? Just a second. So here's your next question. The radioisotope and the technique, I'll talk about this, used to determine the age of the fossil. Let's say you found a fossil and you want to determine the age. Look at the question here. The fossil you found is actually a rock sample. And I'll give a hint here. The rock sample is inorganic sample. So what method you can use to find out the age?
The radiometric dating.
Um,
Carbon dating method.
Carbon dating. Okay. Okay, okay, someone says B. Okay. Yeah, okay. So remember this. This is going to help you in future. Yeah, so the correct answer is B. It is correct. Rubidium 87. Yes. Rubidium 87, rubidium strontium dating method. I said it was a rock sample, which is inorganic sample. So actually there is two methods to determine the age of a fossil. One applies for inorganic sample like stone, rock, which you excavate deep below the earth. And let's say you found an organic, like a mosquito trapped in a rubidium or a stone. So, and you try to determine if that mosquito is from the dinosaur age. So that will be organic sample. On your left side, see the inorganic sample. So for the inorganic sample, the method used is rubidium strontium. And why? It is based on the radioactive decay of the isotope 87, rubidium 87 to strontium 87 by beta decay. So beta decay is the emission of an electron. So rubidium 87 actually decays to strontium 87 and it has a half-life of 49 billion years ago. So thing is rubidium 87 decays to strontium very, very slowly. So this makes rubidium strontium dating technique in principle suitable to date sample from the infant stages of the solar system, from the very beginning of the Earth to the very recent also stone. And how? If you find a rock sample and if you estimate how much strontium 87 is there, so you can reverse correlate or make a reverse chronology to figure out how much rubidium 87 would have been there. And given this half-life of 49 billion years, now you can put in the formula. I'm not discussing the formula, it will be very difficult or complex. So by measuring the strontium 87 level in the rock sample and applying the reverse order, given that you know the half-life is 49 billion years, you can figure out how old the rock sample is. So that is called a rubidium strontium dating method. Since the question asked about the rock sample, the answer is rubidium 87, rubidium strontium method. What if your sample is a organic sample? For that, remember we have radiocarbon dating or carbon 14 dating, not carbon 12. Carbon 12 is the naturally found carbon, but carbon 14, I won't say it is radioactive, but somewhat, let's say it is somewhat higher molecular weight carbon. So it's a scientific method, or you can say it's a radio for like it is higher than the molecular weight. It is not that stable. It is a kind of radioactive carbon. So not carbon 12. Remember carbon 14. Uh, and it can this carbon dating for or carbon 14 dating can accurately determine the age of organic material as old as 60,000 years. This is the limitation. You can also figure out why there is a limitation of 60,000 years because the technique is based. This was, I think, discovered in US by a professor from University of San Diego, I guess. So the technique is based on the decay of the carbon 14 isotope. Actually, it is based on the fact that living organism during their lifespan, they absorb carbon 14 into their tissue. This is the property of carbon 14. It actually gets absorbed to the living organism tissue. When they die, the living organism die, the carbon 14 starts to change into other atoms over time. So scientists can estimate how long the organism has been dead by counting the remaining carbon 14 atoms. So if it would have been dead for a very long time, then minimal amount of carbon 14 would be found. If the organism would have died recently, then like much or more amount of carbon 14 would be found. So these are the two concepts of dating or trying to figure out the age of inorganic sample by rubidium strontium dating or organic sample by carbon 14 dating. Okay. So I hope you people are clear till here. Yes, any questions? Just thing is, I'll be uploading all these slides to the folder and to the link of the folder you can find in your NPTEL dashboard towards the left side. After the class, try to download it and go through it, read it yourself. Any questions, you can mail me. Okay. So these are the two concepts for you. Okay. So since we are talking about the evolution, uh, what do you think? Which cellular organelle is important or is required to trace the evolutionary relationship of an animal? Let's talk about a cell, a organelle. So, what organelle can be used to trace the evolutionary relationship on an animal? Is it mitochondria, chloroplast, lysosome, endoplasmic reticulum? If you know about the reproduction, I think you can answer this question. Somehow you can you have known the answer for a long time. Yes, yes, okay, yes, correct, correct, mitochondria. So if you know that if you have read about reproduction, like the offspring, what the entire uh, mitochondria of the offspring comes from the mother. So the mother contributes to all the mitochondria of the offspring. Correct. So that means, and before going to that, where the DNA is found in the cell. The DNA is found in the nucleus as well as in the mitochondria. So do not forget this, that some amount of DNA, mostly circular, not always, but mostly circular, small circular DNA is also found in mitochondria. Okay. So here have a look. See, if this is the cell, this is the mitochondria. Some small circular mitochondrial DNA is also found. And why I'm telling you this is because the mitochondrial DNA, or it is in short called the mtDNA, has a unique pattern of inheritance. It is directly passed down from the mother to child and it accumulates changes much more slowly than the other DNA. Why? Sorry, why the other DNA accumulate much more changes than the mitochondrial DNA? Because the genomic DNA or the DNA in the nucleus, they undergo recombination. If you would have read this mitosis, meiosis, you would have read about recombination. Recombination leads to variation. So the genomic DNA undergoes recombination, and that is why they have much more higher variation. But the mitochondrial DNA, which is directly passed from the mother to upstream, does not undergo through this recombination and all. They just simply replicate. So that is the reason why they accumulate changes much more slowly than other type of DNA. Because of its unique characteristic, mitochondrial DNA has provided important clues about the evolutionary history. For example, difference in mitochondrial DNA are examined to estimate how closely related one species is to another. This is one example. So if you want to see if species A and B are close to each other, then their mitochondrial DNA or the sequence of the mitochondrial DNA would be very close to each other. So I hope you remember this, that mitochondrial DNA is used for tracing down the evolutionary relationship. The mitochondrial DNA does not mean that simply take the mitochondrial DNA and see it in agar cell. This means that take it and sequence it and see how much sequence similarity is there between one species and other species mitochondrial DNA, not the genomic DNA. Genomic DNA is prone to recombination and variation. Okay, I hope this theory is clear now. Okay, so the next. Okay, since we are talking about mitochondria, we'll talk about what process does not occur in the mitochondria. Is it oxidative phosphorylation, glycolysis, citric acid cycle, or the TCA cycle? Yes. Who remembers from the biology classes? Okay.
Sir, glycolysis, sir.
Yes, yes.
Sir,
Correct,
B.
correct, correct, correct. People who said glycolysis says glycolysis, remember correctly. So mitochondria does not uh, house glycolysis. Glycolysis happens in where glycolysis happens. Yes, anyone who knows where glycolysis happens? Glycolysis happens in the cytoplasm. So the option is B, glycolysis does not happen in the mitochondria. Look here. So this is the entire cell. This is the mitochondria. This is the glycolysis which happens in the cytoplasm or in the cytosol. From glycolysis, glucose is broken down to pyruvate in glycolysis, and the pyruvate then enters the mitochondria. And in the inner membrane of mitochondria, we have the citric acid cycle, and this NADH2 further enters into the TCA cycle or the citric acid cycle. I hope you remember this. So bottom line is glycolysis happens in the cytoplasm, not in the mitochondria. Okay. Just to be clear, do you guys have you people read about this, or is it very new for you? Can someone tell me if this is very new for him or her? This glycolysis
No,
TCA
sir.
cycle? Okay, you people have read. Okay. Just in case if you have would not have read, I would have told a little more about it, but it's fine. These are very basic concepts. Okay. So, glycolysis does not occur in mitochondria. Okay. Okay, so from the starting, we talked about so many organisms. Like I told you, like if we go to a dense forest, you will see so many of organisms, starting from plants, animals, insects, so many things. Actually, everyone, like let's say, if you are telling mango, then the name mango differs from place to place, state to state, country to country. They call it by their local name, or they have their different name. So we cannot rely on the local name or the name given by one person to characterize or identify that particular organism throughout the world. So there is a need to standardize the naming or the name of living organism such that a particular organism is known by the same name all over the world. And this process is called nomenclature. So a number of scientists have established procedures to assign a scientific name to each known organism. This is acceptable. So people uh, unanimously accept this. All the biologists over the world. For plant, there are two organizations that look after or approve the naming. They are ICBN and ICZN. So International Code for Botanical Nomenclature deals with the providing the name of the plants for plants. Or ICZN or International Code for Geological Nomenclature, ICZN deal with the name of the animal or animal taxonomist. So botanical nomenclature is for the plants, geological nomenclature is for the animals. And the scientific name ensures that each organism has only one name to avoid any sort of confusion. Description of any organism should enable the people in any part of the world to arrive at the same name. Like we will see how just us like how the scientific name is formulated or created, so that it indicates the particular organism throughout the world. Okay. Before going to the part of the name, it is actually a two-part system. It has a generic name and a specific name, and the person on the right, he's Carlos Linnaeus or the Carl Linnaeus, who is the father of taxonomist and who designed this. So biologists follow this universally accepted principle to provide scientific name to the known organism. And as I said, it has two components. It has one generic name and a one specific epithet. So it is a genus name followed by the species name. This system of providing a name with two component is called binomial nomenclature, and this naming system given by Carlos Linnaeus or Carl Linnaeus is being practiced by biologists all over the world. The naming system uses a two-word format, found convenient. People found it convenient throughout the world. It was easy for them to name organism based on the binomial nomenclature. Let us take the example of mango, which I was telling you about now, to understand the way of providing scientific name better. So the scientific name of mango, it may vary from place to place, but scientifically or the binomial name, nomenclature name or is Mangifera indica. Let us see how this binomial name is. So the name Mangifera represents the genus, while indica represents the species or a specific epithet. So if you look here, it's written Mangifera indica, M is capital and it is written in italic. None other words are in italic. Only this Mangifera indica is written in italic. Why is that? There are certain rules to write this binomial nomenclature or this two component naming system. So this is the universal rule which is followed. Remember, in case you need to write down these generic names. So the biological names are generally in Latin, and the written, if you are writing it, then they are in italic. They are Latinized or derived from Latin, irrespective of their origin, of their native language or local language. The first word is the biological name, represents the genus, while the second component denotes the specific epithet. Both the words in the biological name, when handwritten, so if you're typing it, it is italic. If you are handwriting it, you cannot write in cursive, right? You cannot make it italic. So if you're you are handwriting it, they are separately underlined. So if I would write in a piece of paper, then I would have to underline it or printed in italics for printing purpose to indicate their Latin origin. And remember, the first word denoting the genus starts with a capital letter, Mangifera indica, a capital letter, while the specific epithet starts with a small letter. It can be illustrated with the following example of Mangifera indica. Okay, but there's a small mistake here. Can anyone figure out what is the mistake here in this Mangifera indica?
Yes, sir. No underline. Not there is no underline.
Not separately underlined, sir.
Okay, no underline. But if it would
Not
have
in Italian.
yes, yes. If it would have been in a piece of paper, handwritten, then underlined. Since it is printed or in a screen, it should have been in italic like this. Okay. So then another thing, like this is not very widely followed, but sometimes followed. Like the name of the author appears after the specific epithet. That is the end of the biological name and is written as like example, Mangifera and indica Lin. It indicates that this species was first described by Linnaeus, Carl Linnaeus himself. So Mangifera indica Lin, capital, italic, and Lin is optional. It is not mandatory. So the author name is at the last. Okay. So now we know how to name so many organisms. But the thing is, we have so many organisms. For example, you go to a library, there are so many books, but the books are organized based on self and on a like which corner of the library or which floor of the library, which makes a person easy to find a book. Because in a library, there is not just one book, there are thousands or lakhs of books. Similarly, to classify or to find out a single organism, you have to kind of make a library, and they should have a particular order, or there should be name of the order so that you can identify an organism. And this process is called classification. So classification is the process by which anything is grouped into convenient categories based on some easily observable characters. For example, we say dog, cat, mammal, wheat, rice, plant, animal, etc. are convenient categories according to us, and we used to study the organism. So, this dogs, cat, mammal, the scientific term for this category is called taxa. We call it dogs and cats. Scientific term is taxa. Dogs, cat, mammal each represent a taxa. So based on the characteristic, all living organism can be classified into different taxa. So this process of classification is called taxonomy. From the word taxa, this process of classification is called taxonomy. So dogs, cat, mammal, wheat. Actually, dogs also belongs to mammals, but dogs is a taxa, mammals is a taxa. Thing is, each organism or each name represent a taxa at different level. There is different label. And what is important for this taxonomic study is the external and internal structure along with the structure of cell, the development process, the ecological information of the organism are all essential and form the basis of modern taxonomic study. Okay, so we will look how they are ordered. This taxonomy is how they are ordered. So that is called taxonomic categories. Okay. So all taxonomy categories constitute the taxonomy hierarchy. So hierarchy is like first the greatest member of the family comes, then the son, then their son, like that. So you can imagine who is at the top is like a more holistic thing or a bigger thing than who is at the bottom. So this constitutes the taxonomic hierarchy. Let us look at the hierarchy. So the bottom one is the species, what we talked about, Mangifera indica. So indica is the species name. Why indica? Because the thing or the organism which form a species which are in a particular species have morphological similarity. Morphology is how they look outside. If anyone does not know, morphology is something which like how they look outside. So it will the lowest is species. And just above it, I said the binomial nomenclature is genus followed by species. So above species is genus. Genus is the aggregates of a closely related species. So I said no, mango is Mangifera indica. So Mangifera is the genus name. Let's go one step above. Above genus comes family. So mango belongs to the Anacardiaceae family. Above this Anacardiaceae family, we have order. Order for the mango is spindles, is the order of the mango. Above that, so as these increases, the category increases, it accommodates a lot of organisms. So Mangifera indica is mango, but Mangifera something else is something other thing. Similarly, this spindles order has so many families under it. Anacardiaceae has so many genus under it. Mangifera has so many species under it, but Mangifera indica is just mango. Similarly, above order is class, like dicotyledony. So dicotyledony is like the dicotyledon seed. So seed is monocotyledon or dicotyledon. So these are dicotyledony forms the class. Above class, we have phylum or division. The term division is used in case of plant. Remember this, the term division is used in case of plant, and the term phylum is used in case of animals. And above all, we have kingdom. For now, I'm telling you two kingdom. In the next class, we'll discuss more about the five kingdom classification. So two kingdom classification is the plant or the animal kingdom. So everything, all the plants fall under the plant kingdom. So plant kingdom. division is angiospermy, and then class, order, family, genus, species. So this is how the taxonomy order has been formed. Yeah, I just in my school days I devised one cheat to remember this because every time you don't remember what comes after, like kingdom, phylum, class, order, family, what comes after what, you might get confused. So I remember from here. So it is I don't know if you guys know or not. Long back there we had PCO phones where you can put coins and make call. So PCO, PCO for girl students. Let's just remember it like that. So it is PCO for girl students. So phylum, class, order, family, genus, and species. That is how you remember this. This is how I remember this. Okay. So I hope this taxonomy categories is clear. Okay. So till here, any question or shall I go to the question? I have few more question and then just one concept, then we'll be done with today's class. All are clear? So we'll proceed to the question.
Yes,
Yes,
sir.
sir.
Okay, great. So the question based on this, the botanical name of garlic is Allium sativum. Again, not in italic. This is a mistake. Allium sativum L. So what L stands for? Is it Linus Pauling, Liliaceae, Carlinus, or Carvin Linus?
Carl Linnaeus, sir.
Okay, okay. Yes, correct. Yes. So people who said option C, yes. People who said option C, correct. It is Carlinus. Not every time you see Allium sativum L because this is optional. The author's name is optional, not mandatory. You can choose not to write it. But Allium sativum, you can write, you have to write with A capital. And since you are writing on a paper, you have to underline it. And if you're typing it, it should be italic. So the option C. Okay. Uh, some few last concept. So I said about evolution of organisms, how one organism or one ancestor started, and from and the one ancestor other organisms formed. So thing is, they inherited from certain characteristic or certain features from their ancestor. And the rules of inheritance of traits. Traits is feature. Simple term for trait is feature. Is given by Mendel. So the on the right side is Mendel. He has contributed enormously to the genetic field of genetics. You must have heard about uh, law of assortment, law of segregation. All these are given by Mendel. I'll just I won't talk about these things now. I'll just give a brief overview of Mendel's experiment and or how come the progeny or the traits or the characteristic feature of the progeny is affected by the parents. So the rule of inheritance of such trait in human being are related to the fact that both, remember here, both the father and mother contribute practically equal amount of genetic material to the child. This genetic material is the genomic DNA, not the mitochondrial DNA. Do not get confused with the mitochondrial DNA. Mitochondrial DNA is only provided by the mother. But now we are talking about the genetic DNA. Forget about the mitochondria. So the genetic makeup of uh, the offspring or the child is by equal amount or equal contribution from the father and mother side. This means that each trait can be influenced by both paternal and maternal DNA. Thus, for each trait, there will be two versions in child. what will then the trait seen in the child be? If there is two versions, one version, so I think all of you know that uh, mammals or the advanced organism, plant, animal, they are all diploid organism. So we have two sets of DNA. Sorry, not two sets of gene. So in the DNA, we have two sets of gene. Each characteristic has two sets. Mendel worked out the main rules of such inheritance, and it is interesting to look at his experiment. So Mendel used a number of contrasting visible characteristic of garden peas. So he chose garden pea for his experiment, and he chose how many parameters? Seven parameters. Flower color, purple or white, flower position, axial flower or terminal flower at the top or at the axis. The stem length, short or tall, seed shape, round or wrinkle, seed color, yellow or green, pod shape, inflated pod or constricted pod, and the pod color, yellow or green. So Mendel used a number of contrasting visible character of garden peas, round, wrinkle, seed, tall, short, plant, and in total he considered seven traits. I'll just talk about the plant height, and tell you about the trait which is expressed or get expressed in the child and how it is inherited from the parent. So in the first place, there was no halfway characteristic in the first. So how he started is he crossbred one tall plant and one short plant. These are called pure lines. That means if you self-pollinate the tall plant, you will get all tall plant. And if you breed this short plant here, you'll get all short plant. So they are called pure line. And as I said, there is a duplicate of the gene. There is two gene for one characteristic. Let's say for the height, we will assign gene T. So for tall, capital T, for short, small t. So all the tall plant, which are pure line, will have capital T, and all the short plant will have a small t. This part is clear. Now he crossbred these pure lines, capital T, small t, and he found that although some plant would have, not some plant, I'll just write down here for you. So this plant will form gamete. Gamete is always formed by meiosis. Meiosis forms haploid gamete, not diploid. Capital T is diploid. It will form a haploid gamete, small t. Small t will form a haploid gamete, small t. Small t capital T will fuse in gametogenesis and will form capital T small t. Since this capital T small t is there, although small t is there, there was no medium height plant or there is no short height plant. All were tall. So what can you tell from this? That capital T is dominant over small t. Although small t is present with capital T, small t was not able to make the progeny plant small, neither small nor even medium height. All are tall. So capital T is fully dominant over small t. Now I'll write it down for it will be easy. Now what he did, he took this capital T small t, that is the heterozygous plant, and self-pollinated. So capital T, small t are made to cross among themselves or self-pollinated. So this one will form a zygote or a gamete capital T, as well as some gamete will be small t by virtue of meiosis because it has capital T small t. Some gametes will get capital T, some gametes will get small t. Same goes for here. capital T, small t. Now let's do permutation combination. This capital T can go and fuse with this one. This capital T can go and fuse with this one. This small t can go and fuse with this one. This small t can go and fuse with this one. Here it is capital T, capital T. Here it is capital T, small t. Here it is small t. Sorry. Here it is small t, capital T. Or I just write capital T, small t. And the last one, where small t and small t fused. It is small t and small t. Now you can see how come they have this capital T, small t. What he observed was only the plant which had small t was short in height, not the plant which has capital T, not the plant which had capital T, small t. So there is complete dominance of capital T over small t, and small t can only express when it is in homozygous condition, when it is present in duplicate. So Mendel called these as a factor, but now we call them as genes. I named these two genes as capital T, small t, responsible for the height of the plant. So in this, I told you about the dominance. So capital T was the dominant over the small t. And so this is the rule of inheritance. By the rule of inheritance, the progeny acquires the character what it is being passed from the parent. If from the parent capital T, capital T is coming, it will be tall. If capital T, small t is coming, it will be still tall. If small t is coming, it will be short. So this is called rule of inheritance, which is given or worked out by Mendel. Okay. So I hope this part is clear. I did not confuse you guys with the capital T and small t. Yes. If anyone wants to ask question here,
Sir, if
so
it is small t and capital T, it is tall or short, sir?
small t, capital T. Yes.
Yes, sir.
So yeah. So you are asking about the plant with small t and capital T. These two, right?
Yes, sir.
Yeah. So it's just the same way of saying capital T, small t, capital T. Both are same. So each organism is a diploid organism. So it will get one capital T, one small t. Doesn't matter what comes first. If it is still the same if it gets small t and capital T. Capital T, small t, and small t, capital T, both are same. So both are tall. And you can,
Yes, sir.
for your simplicity, you can write both as capital T, small t. It will look odd if you write capital T, small t, capital T. First comes capital and then comes small.
Yes, sir.
Okay, cool. So we have one more question. So how many threads were used in Mendel's genetic experiment of Pisum sativum?
7, sir.
Seven, correct, correct.
Seven, sir.
Yes, yes. So garden pea scientific name is Pisum sativum. So seven threads were used. So it is option D. Yes. Okay, correct. Option D, correct. Okay. I think last two, three questions. Uh, a cell is incubated with sulfur 35. Sulfur 35 is a radioactive sulfur labeled methionine. So S35 is labeled methionine. Will label macromolecule X with radioactivity. So what do you think this sulfur 35, what macromolecule it will label? Will it label DNA, RNA, protein, or glycogen? For your reference, this is DNA here, and this is glycogen here. And protein, it already said it is methionine. So what do you think it will label? DNA, RNA, or protein, or glycogen? Sulfur 35.
The protein.
Protein. Okay. Anyone else? I think this experiment you all have would have read. The bacteriophage experiment. It was done by Griffith. Griffith experiment, right? I okay. See this experiment. So the answer is protein. Sulfur 35 will label protein because DNA, RNA, and glycogen does not have or do not have sulfur in it. So it is protein. Oh, sorry, the Hershey Chase experiment. So this one you would have read in your biology books, which is the genetic material. People initially were having the debate, which is the genetic material. Was it the DNA? Was it the protein? So these two scientists, Hershey and Chase, they labeled protein by sulfur 35. So this is how sulfur 35 is used to label protein because it is radioactive. And it is labeled in a red. And this bacteriophage is allowed to infect the cell. And they found that after the infection, only the virus coat is radioactive, not the genetic material which was transferred to the bacteria. It was still not radioactive. But when they used phosphorus 32 or the radioactive phosphorus labeled in green, which is a part, the phosphorus, which is a part of the nucleic acid, DNA, RNA, they saw that the green genetic material was transferred to the cell and the cell was now glowing green. So that is how they argued that DNA is the genetic material and not protein. So this questions comes from this concept, Hershey-Chase experiment. Okay. Uh, if you have time, read about this Hershey-Chase experiment also. Okay. And what protein? So as I said, amino acid make up of protein or the protein are made up of amino acid. So what amino acid has sulfur in it? It is methionine and cysteine. Remember this. In many places you get a question on this. Which amino acid has sulfur in it? So it is methionine and cysteine. These two amino acid has sulfur in it. Okay, one more question. How many number of sister cell will be obtained after three division of a liver cell? Let's say any cell. How many number of cell you think will be formed after three division? So one cell gives two daughter cells. Anyone? How many cells after three division?
Sir, 8, sir.
Eight, sir.
Eight. Okay. Anyone else? You can try. You can write in the chat box. It's okay if it is not correct. Okay. Okay. People are getting there. So where is my cursor? Okay, here. So I'll draw a cell. One cell divides to two cell. This is one division. This divides to two more cell. This divides to two more cell. Second division. Here, this two, this two, this two, this two. 2, 4, 6, 8 total. But let's say if it would have asked you six division or seven division, you cannot take the entire page and draw circles, rounds. Remember this formula? The number of cell after any division, any n number of division, let's say n, is 2 to the power n. So this is the formula. So number of cell after any division is equals to 2 to the power n, where n indicates your number of division. If question is 5 divisions, then 2 to the power 5. If question is 7 divisions, 2 to the power 7. Now you can easily tell how many number of cells are there after each division. On the right side, I put a cell division, the particularly mitosis, A, B, C, D. I hope you all know what is A, B, C, D. Prophase, metaphase, anaphase, and telophase. Then you have your cytokinesis and you have two cells. Okay, here the important thing is remember this formula 2 to the power n. Okay, so the option is A. Whoever said 8, you are correct. Okay, so that's all, people. I hope you like the video, the not the video, the class. So I'll see you next Monday. So I'll be taking, so this course will be having three TA classes. One Friday, one is your Monday, one is your Tuesday. So my TA class will be on Monday. So if you have any question, feel free to ask. Otherwise, you are free to leave the meeting. Thank you all.
Thank you, sir.
Thank you.
Thank you, sir.
Thank you.
Thank you, sir.
Thank you. Okay, so if no one has any question, I'll close this meeting. See you all next Monday. And thank you. Yeah, thank you. Bye bye.